Did you know that every bite of food you take is made possible by invisible engineers, enzymes? These molecular magicians work at lightning speed, making life's essential chemical reaction happen in the blink of an eye. Take honey, for example. It contains an enzyme called sucrase, which acts like a tiny pair of molecular scissors. Its job, snipping sucrose apart into glucose and fructose, making the sugar easier for your body to absorb and turn into energy. Now, let's follow the journey of your honey-covered bread. Once you swallow it, it embarks on an adventure through your digestive system. As it travels, it gradually breaks down until it reaches the small intestine, where the final transformation takes place. Sucrase lives on the surface of microvilli, tiny projections lining your intestinal cells. This animation shows the real magic, how the enzyme sucrase catalyzes the hydroxylase of the common disaccharide sucrose into its constituent monosaccharides, glucose and fructose. When a sucrose molecule lands on sucrase active site, the enzyme reshapes itself, exposing the bond holding glucose and fructose together. Water molecules swap in, breaking the bond and freeing the two sugars. Instantly, sucrase returns to its original form, ready to repeat the process again and again. Enzymes are everywhere, in your food, your cells, and even in industrial applications. Without them, life's chemical processes will be too slow to sustain us. What is enzyme? Enzymes are essential proteins that accelerate chemical reactions in living organisms. Without them, life's processes would occur too slowly to sustain life. Acting as catalysts, enzymes speed up reactions by lowering the activation energy required to initiate them. Activation energy is the initial energy needed to break bonds and start an reaction. Imagine trying to push a boulder over a hill. Enzymes flatten that hill, making it easier for the reaction to proceed. Notably, enzymes do this without being consumed themselves, allowing them to be reused repeatedly. Each enzyme is highly specific, interacting only with particular reactants called substrates. This specificity is often compared to a key fitting into a lock. When a substrate binds to an enzyme's active site, the enzyme undergoes an induced feed, adjusting its shape slightly to create a more effective interaction. This binding forms an enzyme-substrate complex that facilitates the conversion of substrates into products. Enzymatic reactions can be influenced by various factors. For example, increasing the substrate's concentration can speed up the reaction until all enzymes' active sites are occupied. Once saturation is reached, adding more enzymes is the only way to further increase the reaction rate. Environmental conditions such as temperature and pH also play crucial roles in enzyme activity. Too much heat can denature enzymes, causing them to lose their shape and functionality. Conversely, low temperatures slow down molecular movement, reducing reaction rates. Similarly, extreme pH levels can disrupt the enzyme's structure, impairing its ability to bind substrates. Some enzymes require additional molecules, known as cofactors, to function properly. Cofactors can be inorganic, like metal ions, or organic, like vitamins, which are called coenzymes. Conversely, inhibitors can reduce or halt enzyme activity. Competitive inhibitors resemble the substrate and bind to the active site, blocking substrate access. Non-competitive inhibitors bind to other parts of the enzyme, altering its shape and reducing its effectiveness. Cells also regulate enzyme activity through allosteric regulation and feedback inhibition. Allosteric regulation involves molecules binding to sites other than the active site stabilizing the enzyme in either its active or inactive form. Cooperativity, a form of allosteric activation, occurs when the binding of one substrate molecule enhances the enzyme's ability to bind additional substrates. Feedback inhibition helps maintain cellular balance by shutting down metabolic pathways when enough end product has been produced. It's like that in an assembly line factory, the final product raises its hands and shouts, "That's enough! Turn off the line!" Next, we move on to the next section, a detailed demonstration of the BIOL110 experiment, measuring lactase activity. We will explore how to use spectrophotometry to analyze lactase, a key enzyme involved in carbohydrate metabolism. Another enzyme, lactase, is used in the experiment. It is an enzyme that catalyzes the hydroxy of lactose into two simpler sugars, glucose and galactose. In our experiment, we do not use lactose directly, but rather a lactose analog called orthonitrophenol beta-D galactoside, short for OMPG. When lactase acts on OMPG, it produces orthonitrophenol, short for OMP, a yellow compound that absorbs light at about 420 nanometers. This color change allows us to quantitatively measure the enzyme activity using a spectrophotometer. Before we begin, safety is our top priority. Always remember to wear gloves, goggles, and a lab coat when handling chemicals. Now, let's take a look at how this experiment was done. Begin by grinding one lactase tablet into a fine even powder. Transfer this powder into a 50 ml tube-labeled suspension and add 10 ml of PBS to suspend the enzyme. Vertex the mixture for 1 minute to maximize extraction. Distribute the suspension into 1.5 ml centrifuge tubes and centrifuge at around 10,000 rpm for 1 minute. This step helps to sediment the insoluble particles, leaving behind a clear enzyme extract. Prepare several reaction tubes. For the experimental group, mix PBS, OMPG, and the lactase extract flowing in the form below. Additionally, set up control groups, one without the enzyme and one without the substrate, to ensure that any observed color change is solely due to lactase activity. Place the tubes in a dry bath at 37 degrees Celsius and allow the reactions to proceed for varying time intervals. When each time point is reached, immediately add an equal volume of sodium carbonate to stop the reaction. The stop solution not only holds the enzyme's activity, but also stabilizes the yellow color of the product. Finally, pipette a measured volume of each reaction mixture into a 96 well plate and measure the absorbance at 420 nanometer using the spectrophotometer. The absorbance value corresponds to the concentration of OMP produced, which reflects the activity of lactase. Results and analysis. Once the reactions are complete, you will notice that the experimental tubes change color from clear to varying shades of yellow, while the control tubes remain colorless. This confirms that the yellow coloration is the result of lactase catalyzed hydroxylase of OMPG into OMP. The absorbance data collected at different time points provides critical insights. Increasing absorbance. As the reaction time increases, more OMP accumulates, which is reflected in the increasing absorbance readings. Initially, the reaction exhibits a linear increase, indicating a steady production of OMP. Plateau phase. After a certain period, the reaction rate may slow down. This could be due to the depletion of OMPG or potential inhibitory effects from the accumulating OMP. Identifying the linear range is crucial for accurately determining enzyme kinetics. Data reproducibility. By running parallel replicates for each time point, we ensure the reliability of our measurements, minimizing errors from pipetting and timing variations. Through plotting absorbance versus time, we can visualize the reaction kinetics and pinpoint the optimal measurement window for lactase activity. To summarize, the experiment demonstrates that lactase efficiently hydrolyzes OMPG into a measurable yellow product, OMP. The reaction kinetics are directly correlated with time. As the reaction proceeds, more OMP is produced until a plateau is reached. This method not only confirms enzyme activity but also highlights the importance of selecting the correct time frame for kinetic studies. Now that we've explored the experimental procedure and result for measuring lactase activity, let's shift our focus to how this experiment translates into real-world applications. In everyday life, the ability to measure and optimize enzyme activity has profound implications, particularly in the food industry and healthcare. One of the most direct applications is in the production of lactose-free dairy products. For those with lactose intolerance, consuming regular dairy can lead to digestive discomfort. By employing lactase to break down lactose into glucose and galactose, manufacturers can create milk, cheese, and yogurt that are easier to digest. The spectrophotometric assay we used to track the production of OMP is similar to the methods used in quality control processes to ensure that the lactase activity is sufficient to completely hydrolyze lactose. Beyond dairy processing, the principles demonstrated in our experiment are crucial in clinical diagnostics. Assays that measure enzyme activity help in diagnosing lactase deficiency and in monitoring the effectiveness of lactase supplements. By understanding how factors like temperature and reaction time affect enzyme function, healthcare providers can make informed recommendations for managing lactose intolerance. The applications don't stop there. Enzyme kinetics is a fundamental concept that extends into various fields, including biotechnology and pharmaceuticals. Industries that rely on enzymatic reactions such as brewing, biofuel production, and the manufacturing of bio-based chemicals employ similar techniques to optimize reaction conditions. Precise enzyme measurements allow these industries to enhance products' yields, reduce costs, and ensure process efficiency. In summary, the experimental techniques we practiced not only deepen our understanding of biochemical reactions, but also pave the way for innovations that impact everyday life, from healthier food options and improved clinical diagnostics to more efficient industrial processes. This connection between laboratory experiments and real-world applications amplified the vital role that enzyme studies make in advancing science, technology, and public health. At last, I would like to extend my heartfelt gratitude to Professor Zhu-Heng Li, whose detailed and easy-to-understand teaching has truly enriched my learning experience. He even manages to remember the names of all 30 students in just 1 week. I also want to thank my Think Pair Share group members for their insightful discussions that deepen my understanding of biological concepts. Finally, a sincere thank you to my lab instructor for the meticulous guidance provided during my experiments. Your support and dedication have been invaluable. Thank you all. Thank you very much for paying attention to my video.